Okay, everyone, we're going to get started. Yeah, so welcome back from lunch. If everyone can just have a seat. We don't want the day to get away from us, so we can keep on schedule. Just to introduce myself again, for those of you who weren't here this morning, my name is Allyson Berent. I am a veterinarian and a parent of a little girl named Quincy with Angelman Syndrome. I'm also the Chief Science Officer of the Foundation for Angelman Syndrome Therapeutics. And we're really excited about this afternoon session. The focus of the session is going to be on various types of gene therapy or gene editing. And so the next 20 minutes, I'm just going to go through with you a very simple way to think about gene therapy and explain a lot of the questions I think many of you have. I hope to answer some of those so that after me, you're going to hear our keynote speaker, Dr. Jim Wilson, who is going to be talking about gene replacement therapies in Angelman syndrome. So I just want to lay out a groundwork for you to understand the differences, and then we're going to have Gillis talk about gene replacement therapy or protein replacement therapy, and then following that is going to be a panel discussion of all of the key opinion leaders that are working in gene therapy or gene editing at the table, where we've collated questions from parents on social media, and we've organized those questions for our scientists and our clinicians to answer for you. So it's really exciting. Take notes, open your ears, listen carefully, and don't be shy about asking questions. This is your time, and you deserve to have all your questions answered. So just to remind everybody, there's also little, what are they called? Like little picture things out there. There's a photo booth, and then you can also put your face in some frame. Apparently that's the cool thing to do on Facebook, I don't know. So go out there and do it, because everyone's posting them, and that might, you want to join that club of posting pictures like that. Okay. So, therapeutic approaches at our fingertips. This is a really exciting time, and I want everyone in the room to realize that you are really lucky. I feel really lucky that my daughter is in a position that she can potentially benefit from major therapeutics. And those may be upstream, those may be downstream. But I think it's really hard to know how excited you should be if you don't understand why. So what I want to do is really briefly bring it down to, what do these therapeutics mean? What is an ASO? What is a zinc finger? What is gene therapy? A lot of people don't really understand that, and why should you? You're not a geneticist. That doesn't make a whole lot of sense to a lot of us. Unfortunately, I had to get a fake PhD in neuroscience when I got my diagnosis in order to understand what people are talking about. So I hope to just be that translator between the details of the science to what do we as parents really need to understand. And remember, knowledge is power. So listen, understand, ask questions, because you're going to make the best decisions for your kids based on that. So first, what is Angelman syndrome, and how can we fix it? Can we fix it? And I think that's a really important basic question we need to ask. I'm sure a lot of you are wondering, we've raised a tremendous amount of money in the last couple of years. The foundation has worked really hard to raise that money. A huge majority of it is because of the people in this room, if not all of it, or the people listening at home. We work our butts off in a grassroots effort to do this, and you need to know what progress has happened. Where has your money gone? Whether it was $20 or whether it was the unbelievable donation by the Manier-Lapostale Foundation of $6 million. It doesn't matter. You have a right to know where that money went and where it's going and what we're doing with that and why we're so excited. So what progress have we made? What's our agenda to get to human clinical trials? That's why we're here, right? That's what we want to know. That's our mission. So I want to share that with you on where we are. And what do we need from each one of you in the room? And you guys can play a critical role in making this possible and making this a reality. So really briefly, it seems silly. You're a parent or you're a caregiver. How do you not know what Angelman syndrome is? Well, I can tell you I certainly had no idea until I really started to learn about the science. So Angelman syndrome is caused by a reduction in a very unfortunately important gene. That gene is called UBE3A. That gene is one in well over 20,000. It's important. And that gene is found on chromosome 15. There are 46 chromosomes in each of our cells, 23 pairs. We get one of those from our mom and one of those from our dad. So that's called maternal and paternal. Each chromosome is made up of various genes. So we have one chromosome from mom, one chromosome from dad, and there's hundreds of thousands of genes on each one of those chromosomes. And abnormalities in one of those genes can affect a human or any type of animal very significantly. So our children all have in common that they have an abnormality in that single gene UBE3A. And that is a diagnosis of Angelman syndrome, regardless of how that abnormality happened. So if we look at chromosome 15, that's 2 copies, one from our father, one from our mother. And if we look at that in a schematic, what you see is our father's copy and our mother's copy is harboring UBE3A right here. But something really, really fortuitous about the 15th chromosome and the UBE3A gene happened. And that is something we can really use and manipulate. So what happened is the father's copy of that gene has been silenced, and that's called imprinting. So I'm just going to make this really simple. Let's pretend that someone put a stop sign over it so it can't be read. So if you're driving a car and you're going down the chromosome to read all these genes, you're going to divert and go around UBE3A and then continue to read the rest of the genes. It's a little more complicated than that, but let's just keep it there. The maternal copy is normally read. So we have full reading of the maternal copy, including the gene UBE3A. So if there is a problem with the UBE3A gene on the maternal copy, what's going to happen is that we don't have a readable copy. Most genes in our body, we have 2 readable copies. And so if there's a problem with one, you probably don't know a whole lot about it because the other one is functioning and taking the place of that. Unfortunately, with Angelman syndrome, that's not the case. Because we have a stop sign sitting over the paternal copy or the father's copy, the mother's copy is not being read. So that leaves us deficient in this gene and the functions this gene provides. So what does that mean for your kid? Well, a lot of kids have a deletion, a maternal deletion, meaning a segment is missing. Okay, so that's about 70% or so of our children. Some of our children, so maybe 5 to 7%, have 2 copies from the father called UPD. 2 copies from the father means that there are 2 stop signs. So they're not missing their gene at all, but they have 2 stop signs, so it's not being read. Other kids have a mutation, so the gene is there, but there was a base pair, so a mutation in one of the pieces of DNA that was abnormal, so it's not being properly read, so it can't make a proper protein to function, which is what genes do. And then the other is an imprinting center defect, and that's something that's rare, about 3% or less. So that's a very similar situation in that it's present, but it is not being read properly because of a center defect. Okay, so the result is we all have something in common. We're missing a functional UBE3A gene. So why is that so important? What does UBE3A do? Why is a small microdeletion or a mutation so important? Well, the gene does a whole lot. And this is a picture I stole from Therapeutics, so I thank them in advance for that. But this picture, when I first got my diagnosis, really resonated with me. This made me understand what my daughter Quincy goes through every day. So the gene is vital to how the brain functions, how it controls various things like speech or movement or learning and memory. It provides instructions on how to make a protein, and then that protein is called a ubiquitin protein ligase. So it's an enzyme. That enzyme is very functional. That functional protein is required to control the number of other proteins that are in the neurons or in the brain. So the brain cells are neurons. So if we control those, what happens is if we don't put an enzyme on some of those proteins, too many of them might be stuck around because the enzymes break them down and help metabolize them. So basically it's not allowing this control to be normal, so we have too many of some proteins and not enough of other proteins because we don't have this enzyme that's controlling that environment. And there's likely hundreds of proteins that are being affected by this gene. So if the protein is not removing some of these other proteins in the brain, then we have a buildup. And that buildup might result in an excessive amount of what we call noise in the brain. So an example of that, which Ovid gave earlier, is that we have 2 neurons that meet and they talk. So this is one neuron. The junction between the second neuron is called a synapse. So that junction is very important. And the first neuron releases chemicals to tell the second neuron what to do. And if you don't release those chemicals properly, the second neuron is lost. And so what can happen is that one of the most beneficial effects of the chemicals that are released here, they're called neurotransmitters, is one called GABA. Now, GABA is just one example, but I'm using this example because I think it makes a lot of sense. That one example ultimately means that if we don't have that enzyme, ubiquitin ligase, that was made by the UBE3A gene, then one of the proteins is not broken down, called GAT1. GAT1 is a protein receptor. If that's not broken down, we have too much of these blue channels. These blue channels are responsible for eating up GABA. So if we have too many, we have a loss of GABA. If we have a loss of GABA, that results in symptoms. What are those symptoms? Well, GABA is responsible for what we call tonic inhibition. So a lot of you have heard that word. We throw that around a lot in the community, tonic inhibition. What does that mean? That means that if I'm walking, I can tell my left foot to take a step and let my right foot relax. But then I want to walk with my right foot, so I'm going to tell my right foot to activate and my left foot to relax. If I want to talk, there's hundreds of muscles in my face and my tongue and my throat. Some need to relax and some need to activate. And if I don't have that relaxation potential and that activation potential coordinated, what do we have? We have too much excitation. We're too excited. We don't have tonic inhibition. We're not relaxing. So what does that leave? Poor balance, poor motor coordination, a lack of speech, seizures, poor sleep, anxiety, dyspraxia, apraxia, poor attention spans, and a learning disability because there's so much noise. It's like windshield wipers going on all the time in the rain, and we have to try to see through that, and that's really hard. So I don't know about you guys, but basically that describes my daughter. So she doesn't have enough tonic inhibition. Her body, her brain is too excitable, so she can't figure out how to relax and activate at different points that's appropriate. And that's what Angelman syndrome essentially is, to keep it simple. So what can we do to fix this? And I think if you don't understand that, you're not going to understand what we could do to fix it. So this is a neuron, okay? So here's a neuron in the brain, which is a brain cell. And this is one neuron talking to another neuron, okay? That's the synapse that we were just talking about. So now there's our 15th chromosome that's sitting inside the cell body or the nucleus of that neuron. And that's very important, and that's where all these proteins are being made. But we have a stop on the maternal and a stop on the paternal because we're missing our maternal and we're stopped on our paternal. So what can we do to allow for function of that gene? Well, one thing we could do is we can replace it. Why don't we give the gene back? Okay, that seems really simple, right? Well, if it were so simple, then when this all started about 30 years ago, it would have already been done. It's not simple. It's very complicated, but not impossible. And that's where we are today. So we can do that by delivering a gene or delivering the protein that the gene makes into the cells of the central nervous system or the neurons. How do we accomplish that? Well, we need a vehicle to deliver them. So that might be with a virus. Now that sounds really scary. We're going to put a virus into our kid's brain. How do we do that? Well, that's what we're going to talk about later this afternoon. But it's a very healthy virus. So it's a virus that all the negative things, all the infectious parts of it have been taken out so that they shouldn't get sick from that virus. It's used as a vehicle or a car to drive the gene or the protein into the cells of the brain. So a virus, we manipulate it for it to be good at what we want it to be good at, which is infecting, or we call it transfecting cells. So we can take that virus and we can use one that might be really specific for a neuron. It can get into that neuron. When it gets into that neuron, we could maybe manipulate it so it releases, instead of the viral genes, it releases the UB3 gene. Well, how amazing would that be? We use a virus, and instead of it releasing something bad, we have it release something really good, what our kids is missing. That's gene or protein replacement therapy, to keep it simple. The other option is turning on the paternal allele, or stopping the stop, or unsilencing the father's copy of the gene that's been silenced. So that's turning on the paternal. How can we do that? Well, we can do that with things like artificial transcription factors. We could do that with things like ASOs, or antisense oligonucleotides. And we could do that with various other more novel methods like CRISPR technology. So what I want you to focus on here is that when we talk about viral delivery, when we say gene therapy, that's what we mean, gene replacement therapy or protein replacement therapy. When we talk about gene editing, this is what we mean, turning on the paternal copy. Ultimately, it's all under the umbrella of gene therapy, but I want you to know when we speak of gene therapy, most people are talking about viral delivery, replacing the missing gene or protein. When we talk about turning on the paternal, we talk about stopping that imprinting and activating the father's copy that's already there and healthy. So I just want you to understand that. So when we hear about all these different pharmaceutical companies and biotechs and different universities and different groups that are working on therapeutics, you have in your head these names, but you don't know where they fit. So I just want to share with you where they fit. Okay, so University of Pennsylvania, which is Dr. Wilson, who will be speaking next, he's working on adeno-associated virus delivery of genes. Agilis, who will be speaking after him, is working on also gene or protein replacement therapy, adeno-associated virus delivery of a gene or a protein. So they're going to be different approaches, but they're subtly different, but they could have miraculous differences for our kids, so we have to realize they sound similar, but they're very different. Lentivirus, a totally different virus, lentivirus, okay? That is something that UC Davis is working on with hematopoietic stem cell therapy. We're going to talk a little bit about that. Artificial transcription factors, UC Davis. We also talk about that as zinc fingers, same thing, okay? Zinc fingers, artificial transcription factors. And then the ASOs, or the antisense oligonucleotides. Well, we've all talked about that for a long time, thanks to Dr. Arpodet and Ionis. And now today, which I don't think many of you registered, we just heard that Roche announced that they're also in this space. They also are developing an antisense oligonucleotide for Angelman syndrome. So this is amazing. We have all of these people that have now come to the table to bring various types of gene therapy or gene editing to our kids to either replace or activate the gene, okay? Then there's downstream therapeutics. So downstream therapeutics means what did that gene do? We talked about the GAT, the receptor and two little GABA. So that's what Ovid Therapeutics talked about. So they're going to replace the GABA, and they're going to replace one of the strongest GABAs that's out there to really impact the GABA effect and increase tonic inhibition. That's not a gene therapy. That's not a gene editing. That is a downstream therapeutic that could have significant impact on the symptoms that our kids have. Okay? Great. Okay, so those are the things we're going to focus on for the rest of the afternoon. So current therapeutic potentials, gene therapy, like I said, replacing the gene that's missing. This was published in 2011, thanks to Dr. Weber and his group at the University of South Florida. Dr. Jen Daly, who wrote this paper, is in the room and is going to be speaking to everybody next. So they used adeno-associated virus in mice, the first time this was ever done, to replace the gene that was missing in mice. And miraculously, it was successful. And so that was the first time that people realized that therapeutics for our kids could really have a significant impact. And so what they found was that they were able to rescue, using a virus, various different cognitive defects in adult mice with Angelman syndrome. So that was very, very exciting. What about turning off the antisense and turning on the paternal copy of the gene, or unsilencing it? Well, back in 2011, Dr. Philpott at the University of North Carolina, he published that he could activate the paternal copy using a chemotherapeutic agent called topotecan, or a topoisomerase inhibitor. That was groundbreaking. Somebody can actually do this, and a drug can turn on the paternal copy. Huge news, very important information. And soon thereafter, Dr. Baudet's group at Baylor College of Medicine, they looked at antisense oligonucleotides to essentially do what topotecan did, but in a much more sensitive and specific way to not harm other genes in the genome, but just affect the father's antisense transcript. And so that was published in 2015. And then Dr. Dave Siegel at UC Davis published using artificial transcription factors to do exactly the same thing in a specific way. Okay? So this is the ASOs going from no activation to activation of the paternal copy. So really, really exciting. This is zinc fingers, or artificial transcription factors, going from very little signaling to very dramatic signaling. So that's Dr. Barb Bayless's paper. She's also in the room. So everybody that's been working on this in Passionate is in the room because they believe and want to see this through, that this is going to happen for our kids. This is a time that has never been so exciting. And the last approach here is stem cell-associated gene therapy. So this is something we haven't talked a whole lot about in the community, and next year I think we're going to talk a lot more about this because this is very preclinical. But I want you to know where your research dollars are going. We started this program looking at stem cell delivery of lentivirus delivery through stem cells, and I just want to share with you another disease called metachromic leukodystrophy. So metachromic leukodystrophy is a degenerative disease where kids basically get almost like an ALS syndrome. So their brains get an accumulation of proteins and their neurons die. It's degenerative, and they're born normal. By the age of about 4, they start having some symptoms, and by the age of 6 to 10, their brain is highly degenerative, and it's fatal. So these kids had this gene therapy done through hematopoietic stem cells, and if you look here, this is before gene therapy, when this kid was 16 months old. This is after gene therapy, when this kid was 39 months old. If you compare the difference in the brain on an MRI, if you look here, there's very little difference. If you look at the untreated 39-month-old child, you can see all the black, which is basically a loss of brain cells. So the dramatic change that gene therapy had on these children was so incredible that this is why we decided to fund this type of research as well. So it's another gene therapy, delivering it through a different virus, through a different mechanism. And I just want you to feel educated about that when you start hearing about that in the next 6 months. Okay, so each therapeutic target can benefit our kids in different ways. Investigating each option very thoroughly, trying to find the ultimate symptomatic cure for various different areas of our children and their needs is very, very important. Proof of concept to clinical trial to success is what we want. And we have to take our time to do that in the most safe, in the most efficacious, and getting the best people to the table that have nothing but respect for our children and for our families. And that's what we've tried to do here. So you wonder why it takes so long, but it's not easy. But just to show you, we're going to be done in a minute, is that 1 year ago when we presented, this is where we were, okay? Adeno-associated virus was right here. So if we look at the trend to go from proof of concept, animal testing, human clinical trials to an approved drug, okay, we were here with AAV 1 year ago. Okay, ASOs were here. So we were getting close in that preclinical arena, animal testing, 1 year ago. Zinc fingers just behind that. Gaboxidol, or OV101, and the ketone esters were talking about clinical trials, but they hadn't started yet. They were approaching the FDA, so they were close a year ago. And various things like stem cell therapy were very proof of concept. They were not anywhere near preclinical testing. So where are we now? Okay, this little red box is what we need to fill. So this is where we were, this is where we are now. That's a huge step in 1 year. And I want you to feel what we know is that this is really huge, and to put it in simple terms and not medical and not scientific terms, this is huge. And a second approach is right there. So we're getting there, we're so close. ASOs, we were there, now we're here. Zinc fingers, we were there, and now we're here. Gaboxidol, OV101, we were there, and now we're here. Look at this, we're in phase 2 clinical trials. We were not even in any clinical trial 1 year ago. Thank you to Ovid. And stem cell therapy, we were there, and now we're here. So we have made so much progress in 1 year, it's not fair for us to know that, and for you guys in this room to not know that. And I want you to just be so proud of what your money has done for all of our kids, because it's incredible. I said I wouldn't cry because I'm going to save that for tomorrow night. So where are we now? Forecasted agenda, 18 to 36 months. This could be in human clinical trials, one or some of these approaches. This is for real. This is not lip service. This is for real. Gene replacement therapy, protein replacement therapy, activating the paternal in various different ways. at the FDA or first in human is real. It's going to happen, and we need to be prepared, and we need to know and have knowledge, because it's our responsibility as parents to do that. So what can you do? Obviously, we need money. I'm not just here to raise money, because it's for my kid too. I have a horse in this race. We need to raise money to get through biomarkers, outcome measures, funding these studies, getting through this investigational new drug approach so that we can get to that first-in-man time. That all costs money. That's being funded by many of you in this room. So every $20 counts. This is something that you can help with. Register the global registry that Megan and Chloe talked about this morning. Hugely important. We need data. We need to know how our kids are changing. We need to be able to track our kids. We need that information. So go home. It takes no more than about an hour, hour and a half. Go and do it. We need to have this full. Participate in studies. The ABOM is really needing outcome measures to prove that these therapies work, or the FDA will take it away. So if it's working a little bit, but we can't prove that, it's done. We need to have the right measures so we know what we're testing and we can show changes. And prepare for great things, and that's what you could do for your family, for your kid, and for yourselves. So we unite for a cure. That's what FAST is doing. That's what we aim to do with everybody, every player that's here. Whether it's the different foundations, whether it's the different institutions, whether it's the biotechs and the biotherapeutic companies. Whatever it is, it's all for our kids, and we're here to fight for a cure. So let's make it happen, let's get excited and pay attention, because this is happening very, very quickly. Obviously, we need to thank everybody for making this possible, but from the bottom of my heart, the Fast Fire team are a group of scientists that never stop. They work day and night for our kids, and I want you guys to realize that they may speak on a level that you don't always understand, no matter how much I tell them to tone it down a little bit, but it's only because they're really excited, because they are making breakthroughs every single day, and they can't wait to tell you every year all the great things that they're doing. So for me, our team is so instrumental in this. So I feel like we need a round of applause, not only for them, hold on, but for the Meunier-Lapostale Foundation, who made this leap. This would have not happened without them. So I am so grateful to everybody involved in this community, but for the Fast Fire team and for the Meunier-Lapostale Foundation, I think we all deserve a round of applause. All right, that's all I got. I can take some questions, or we can just keep going since I think we're a little bit behind. And there's going to be lots of questions at the end, so I think a lot of your questions are going to be answered. Oh, sorry. That's my life, okay? So I may have a kid with Angelman syndrome, but my husband and I have a wonderful life, and I hope all of you enjoy your life, because no matter what it is, it's how you make the best of it. So we do have a wonderful life. So I'm going to change over to Dr. Wilson, so I can take some questions while we're changing over.
Hello. Quick question. So are the paternal genes in mice for UBE3A, are those silenced as well?
Where's your there? You want to put it on the hard drive?
An Angelman syndrome mouse, multiple Angelman syndrome mice have been made thanks to Dr. Art Baudet and Scott Dindo and Ben Philpott. So there have been various different types and like strains of Angelman syndrome mice that have been made. So yes, they are typically a mutation or a deletion mouse. So they do not have a functioning maternal copy, and they have a silenced paternal copy.